Pseudoreplication is a very common problem in statistics, and chances are that also you have fallen into the trap of using pseudoreplication. In 1984, Stuart Herbert published a study in which he reviewed a number of ecological studies and found that almost half of the papers using statistical tests or similar analysis were done with pseudoreplicated data. And even decades later, pseudoreplication continues to be a common pitfall. In 2010, a paper reviewing articles in neuroscience found that pseudoreplication was used in 12% of the papers, and a further 36% were suspected to have pseudoreplicated data. So what is pseudoreplication and how can we avoid it? Imagine you want to compare the number of insect species in a field treated with a chemical or a fertilizer compared to a control field which is untreated. Maybe you have placed 10 traps into each field to count insects. What do you think? What is your sample size? If you think your sample size is 10, then you have fallen into the trap of pseudoreplication. When considering sample size, we have to focus on the experimental unit. Here the unit is the field. We have one treated field and one untreated field. Hence the sample size is 1. If we measure several times in different places of one field, the sample size is still 1. It is important to note that these 10 samples per field are not independent, because they are in the same field. The field is the experimental unit, and possibly one field is slightly drier than another one, or it has a different soil structure or inclination. Whatever may be particular to this field, it will affect all samples in that field. So the samples are not independent. Having several samples per experimental unit still helps, because you will obtain a more precise estimate for this field. This leads to smaller variability between samples, between different fields, and hence you will have a good statistical power. But the sample size, in this case here, is still 1. So how would our test design need to look like in order to avoid pseudoreplication? We would use 10 different sample locations, and each one would be treated individually. So basically, we would have 10 fields in the control and 10 in the treatment, and in each field there would be just one measurement. Let's look at another example. Imagine you have 2 fish tanks with 30 fish per tank. One of the tanks is treated with a chemical and the other one is untreated. You measure fish growth. So what is your sample size? Well, now you know it's not 30 fish. The sample size is again just 1, because the experimental unit is the tank and we only have one tank per group. To achieve a sample size of 30, we would need to use 30 tanks per group. But maybe you only have 10 tanks. What can we do? You could use a sample size of 5 tanks per group, and in each tank you could keep 3 fish. This way you could use the average of the 3 fish per tank to obtain a more precise estimate of the mean fish growth in each tank. This will reduce variability between the experimental units, the tanks, and this way having a small sample size of 5 tanks per group may still allow you to detect even small differences. Pseudoreplication is also problematic because it can be used to manipulate the results. Let's illustrate this in the following example. Let's assume we have 3 fields. These are the experimental units. The sample size is 3. In each field we install 6 traps to collect insects. Here are the data that we collected. Now the correct way to test if there are differences between treated and control fields is to use the mean value for each field, because our experimental unit is the individual field. If we consider these data and conduct, for example, a man-with-new test, then we find that there is no difference between the 2 groups. However, if we pseudo-replicate these data, assuming that the 6 values from each field would be independent, which they are not, then we would have a sample size of 18 and our p-value would be 0.032. Of course, this result is simply wrong. There is no significant difference here. But you can see how one could manipulate results when using pseudo-replication. Depending on your data, you could also hide a true significant difference when using pseudo-replication, so just make sure you never pseudo-replicate your data. In general, as pointed out by Stanley Lassick in this paper from 2010, pseudo-replication results in unrealistic confidence intervals. This is the confidence interval when using pseudo-replication, and this is the true confidence interval of the experimental units. And second, pseudo-replication results in unrealistic, or maybe one should better say in wrong p-values. So make sure you never pseudo-replicate your data. Thank you for watching, and if you liked this video, please like, share and subscribe.